I'm going to talk about gene profiling for new biomarkers for Aspergillus. I'm a molecular biologist in infectious diseases. I'm used to designing and developing diagnostic tools for detection of viruses, for example, avian influenza, and using molecular tools for genotyping viruses, parasites, and bacteria. When we have a positive result, that's quite strong evidence that there's a pathogen present and that the patient needs treating. Fungi are a different kettle of fish entirely, I'm discovering, because fungi are present in the environment. So using a DNA-based molecular assay tells us that fungi is present, but it doesn't necessarily tell us whether that's invasive or not. Hence, I've posed the question from the research side of things to determine whether detecting nucleic acid, but using RNA, can confirm invasive aspergillosis, because Aspergillus is in our environment, and I'm assured that if there's building works, you're guaranteed to be exposed to Aspergillus. I'm going to speed through the introduction and current methods of diagnosis because that has been covered quite well this morning in the talks that we heard already. Invasive fungal infections cause 50 to 90% morbidity and mortality in immunocompromised patients. And the frequent organisms associated with that are Aspergillus and Candida. Diagnosing it can be based on clinical suspicion, culture, which is quite slow and not necessarily reliable as a tool, tissue biopsy, which is invasive to the patient and not ideal procedure for a thrombocytopenic patient, for example, and radiological findings, which can appear quite late. So with a late diagnosis, and that delays treatment, causing a worse outcome for the patient. Uncertain diagnosis can cause us to use empirical antifungal drugs, which is costly. And either delay or uncertain diagnosis can increase the length of stay of patients in hospitals, which is again costly. This table just covers some of the antifungals that are available to us. It's by no means extensive because I've not included voriconazole, for example, and I appreciate the costs are different between trusts. But just as an idea, it's possible to see that if we have suspicion of a systemic fungal infection, often the go-to drug is ambisome. And if we go back to the multiples of 7, 14-day treatment will cost over £7,000. So it's important to have appropriate diagnostics in order to proceed with useful treatment for the patient. Patients that are at risk of invasive fungal infection include the following: transplant patients, patients on immunosuppressive therapy, HIV patients, neutropenic patients, and premature neonates. But these are just examples. EORTC have defined what invasive fungal disease is, and one can use host factors, clinical evidence, or sometimes microbiological evidence to determine whether a patient has possible, probable, or proven IFD. This hasn't come up properly on this slide, but microbiological evidence, sometimes we rely on it, sometimes not. And it would be this key area that I would like to address with this talk. By using a more rapid and informative diagnostic tool, we'd be able to have earlier and targeted intervention, reducing mortality, and we'd be able to narrow the differential diagnosis in complex septic patients. We'd reduce empiric antifungal treatment and choose more appropriate antifungal drugs, improving epidemiological data along the way. So current laboratory biomarkers that are in use as mentioned, galactomannan, it doesn't require a huge volume of sample. It's rapid. However, it does produce false positives when patients are on antibiotics, for example. The beta-D-glucan assay is very good. It's a pan-fungal assay as opposed to the galactomannan, which is specific for Aspergillus. The beta-D-glucan, again, only requires a small volume of sample. It has a high negative predictive value, as we've heard already, and again, a very nice rapid assay to use. However, it is pan-fungal, so it's able to detect a multitude of pathogens, excluding zygomycetes, basidiomycetes, and cryptococcus. There's also the lateral flow device, which is possibly going to be readily available to a lot of the trusts, and that's Aspergillus specific as well. From a molecular point of view, there are DNA PCRs. The 18S Aspergillus consensus PCR was assessed or evaluated across multiple sites in the UK. And there's one can use a 28S pan-fungal PCR as well. This is a gel demonstrating that when 28S pan-fungal PCR is applied to culture-grown material at 24 hours and 80 hours for Aspergillus fumigatus, it produces the expected band size. The amplicon has the same intensity across those two time points, so it isn't easy to distinguish between those two time points. So my first question for you is, what microbiological methods do you use locally? A, culture, B, beta-D-glucan assay, C, galactomannan assay, D, consensus Aspergillus PCR, or E, Candida PCR. You can choose a multitude of these except for F, which is all of the above. So if you'd like to vote. Wow. Okay, so the majority of you still incorporate culture techniques, and then there's the galactomannan assay. But there isn't such a lot of you using the PCRs, for example, or the beta-D-glucan assay. So if I review the table that I showed earlier and include the resistance column, this is by no means a reflection on the antifungals themselves. It's more to show that when we're using pan-fungal assays, for example, they don't actually tell us what organism we're dealing with. Therefore, it's difficult to know whether the antifungal will be effective in treating that patient. One certainly wouldn't use fluconazole, for example, for Aspergillus. And Aspergillus terreus is, I believe, intrinsically resistant to ambisome. So it's important with a new diagnostic tool to try and decipher exactly what organism we're dealing with and therefore make the treatment more appropriate. I'm going to now talk about the preliminary data I have from the research and development that I've been carrying out over at King's College Hospital. I really wanted to know whether Aspergillus fumigatus is invasive or not when applying an RNA RT-PCR looking at gene expression levels. I just want to point out that this is self-drawn, so you can probably see why St Martin's Art College have never contacted me to study there. So this is to summarize what my research approach was. I used protein expression for one approach and gene expression for another approach in order to identify targets that might indicate whether they would be useful for determining gene expression levels in early or late or non-invasive and invasive Aspergillus infection. I set up a model system which involved using Aspergillus fumigatus and inoculating that into human fibroblasts, lung fibroblast cells. So that's the first flask there. Then I had a flask for the MRC5 cells on their own, Aspergillus fumigatus on its own, and media. And I collected the supernatant for extracellular protein expression work, and I harvested the mycelium cells and cells for RNA extraction. And this was done at 6 hours, 24 hours, 48 hours, 72 hours, and 80 hours. This is an example of what the MRC5 cells look like when they're uninfected, and then when they've been infected with Aspergillus fumigatus at 24 hours. And as you can see, there are mycelium present already at 24 hours. The Aspergillus in media on its own formed bead-like spores compared to Aspergillus in cells, which had an elongated spore formation. So at a macroscopic level, one can already see that there is a difference in the development of Aspergillus in different environments. So with the supernatant, with these extracellular proteins, if I take this by lane, this is Aspergillus plus cells, a ladder, cells and Aspergillus, and that's at 6 hours. And then Aspergillus plus cells, Aspergillus at 24 hours. And the protein band patterns were very similar between those time points. But when you fast forward to 80 hours, Aspergillus plus cells showed distinct protein bands that were not present in the earlier time points. I took those protein bands, characterized them using mass spectrometry, and identified serine proteinase, which is involved in tissue invasion, and alkaline phosphatase, which is a hydrolytic enzyme. So these were two target regions that I used in order to design and develop an RT-PCR. Now, for those of you less familiar with real-time PCR, I just want to point out that this is, on the axes are the number of cycles of amplification, and ordinarily that would only go up to 40, but I've chosen to go up to 50 cycles. And on the y-axis is the fluorescent signal emitted from the probe that's in the reaction. This is the threshold level, and when this trace crosses that threshold, we can calculate the cycle threshold value, the CT value. By the lower the CT value indicates the higher amount of RNA present, and vice versa, the higher the CT value indicates the lower amount of RNA present. So in this example, I've taken triplicates of the 24-hour sample, which is this line here, if I can keep my hands steady enough, and then I've taken triplicates of the 80-hour sample, which is this line here. In other words, the 80-hour sample demonstrates that there's more RNA present than in the 24-hour sample. Now, a 3 CT difference in values indicates a log difference, and that's deemed significant. I'm not going to ask you, as one of the questions, to do mental arithmetic and give me the average of the CT values for the triplicates, because I've already calculated that on the next slide. But I'm mindful of the fact that the people at different laboratories have different access, and therefore not everybody has access to real-time PCR. So I ran the amplicons on a gel, and as you can see, for the 24-hour sample, 80-hour sample, all of the amplicons appear to be the same. They have the same intensity. This is the calculation of the CT value when averaged. So for 24 hours, that had a CT value of 39, and for the 80-hour sample, it had a CT value of 36. The difference between these two time points is deemed significant because there's the 3 CT difference. I also included a fungal reference gene so that then I could say whether it was relative gene expression to the target gene. So in other words, the target gene here in relation to the fungal gene showed a 6 CT difference, which is a 2 log difference in the 24-hour sample, whereas the 80-hour sample only had a 1 CT difference, which is not significant. In other words, the RNA being expressed is similar to the RNA expression level with the fungal reference gene. These two targets are very helpful and very useful, but I also wanted to look at the gene expression level using next-generation sequencing. By taking the 2 time points, 24 hours and 80 hours, I managed to obtain good coverage. In other words, 28 million reads were obtained per base for the 24-hour sample, and that was higher in the 80-hour sample. I needed a quality score above 30%, and it was 92% and above for both time points. From doing that work, I managed to identify 9,982 gene regions, 12 of which were in the 24-hour sample, and they appeared to be downregulated genes, and 31 genes in the 80-hour sample, which appeared to be upregulated. I haven't looked at all of them yet, but from some of the work that I've done already, I'll just explain the up and down regulation first. In the 24-hour sample here, compared to the 80-hour sample, this gene region is highly expressed, whereas here it's not, and that's classed as downregulation, and vice versa. In the 80-hour sample, this gene region was highly represented, whereas down here it's less so. This is an example of target regions where genes were found to be differentially expressed. There's a transfer RNA leucine that at the 24-hour time point, there was high representation of that gene, whereas at 80 hours, there wasn't that gene was not represented. What I found interesting was the fact that transfer RNA for serine was present, and as mentioned, serine proteinase was one of the protein targets that I was using for designing an RT-PCR. So the second question I have for you is, what is rapid diagnosis using microbiological methods for you? Do you think 3 to 4 day turnaround time is suitable, 7-day turnaround time, a 1-month turnaround time, or none of the above? If you'd like to vote. That's almost dichotomous. So none of the above has the highest percentage there with 49%, and 3 to 4 day turnaround time is considered good by 44% of you there. 7-day turnaround time seems acceptable for 7% of you. So back to the story. I just developed an RT-PCR using some of these next-generation sequence gene targets, and I compared the 24-hour sample in triplicate to the 80-hour sample. Some primer sets did not amplify, to be expected, and some amplified equally in both time points, but some primer sets showed clear upregulation. And this is an example of that. The 80-hour sample was present at 27 CTs, and the 24-hour sample was present at 38 CTs. That's more than a 10 CT difference between the two time points, which is a 3 log difference. That's a significant difference, and very useful for us being able to differentiate between the RNA present in those time points. It's not easy to see, but there is an outlier here. This triplicate is nicely bunched together, whereas we only have 2 of these bunched together in the 24-hour sample, and this outlier here. So I'd just like to show you that on a gel. That outlier is this amplicon here. I'm going to require more time to look further into this to find out what the primers are binding to potentially, but this amplicon here is the expected band size. And as you can see, for the 24-hour sample, the amplicon is not as intense as you see with the 80-hour sample, which is if one doesn't have a real-time PCR machine, the conventional method can indicate that there is a difference between those time points. So if we take this from a clinical point of view, you have a patient that is admitted to hospital on day 0. They're there for maybe a transplant procedure. And a few days later, maybe even after the transplantation has taken place, there's clinical suspicion of invasive fungal infection. This assay, even in its current state, would be able to distinguish between those 2 sample types, 2 time points when sample from day 0 is compared to sample day 3, 4, 5, 6. I used a reference gene, but on this occasion I used a human reference gene. And by doing so, that impacted on the assay itself. So the same samples, the 24-hour sample and the 80-hour sample, were compared. And we're talking about exactly the same batch. But with the reference gene, there appears to be some level of competition in the reaction. And therefore, only the 80-hour sample was amplified. This is the representation of the reference gene, which is handy to see because if we're dealing with patient samples, we need to know that there hasn't been an inhibitory effect in the reaction, and this makes it a lot easier by having a human reference gene present. On a gel, again, there's no amplicon visible for the 24-hour sample, but an amplicon visible for the 80-hour sample, which marries up quite nicely with the real-time PCR results. I'm showing you here the target gene compared to a human reference gene and 2 separate fungal genes. For the 24-hour sample with any reference gene, there was no amplification, there was no CT value for that sample time point. But for the 80-hour sample, there was a 2 log difference when a human reference gene was used, a 1 log difference for either of the fungal genes used. Now, this is demonstrating what it would look like for an upregulated gene to be included in an assay. I would also like to include a primer set and probe for the downregulation, so then it's a more comprehensive RT-PCR diagnostic tool. So in summary, improvements have been made recently in the ability to use biomarkers for the detection of fungi. I've managed to identify potential targets for the development of a specific invasive fungal infection RT-PCR for the diagnosis of Aspergillus. The differentiation between early and late time points was achieved, resulting in the development of an invasive Aspergillus RT-PCR. For the future, I'd like to optimize this assay and apply it to clinical samples, so that then I can determine what clinical impact that has and whether there's a cost effectiveness of using biomarker assays. I'd like to improve the reliability of diagnostic-driven targeted treatment by developing an informative, standardized RT-PCR for early and specific detection of common invasive fungi. In that way, improve antifungal stewardship and patient management. Further research will take place for pathogen-host interaction, which could also be added to a panel of diagnostic tools for investigating invasive aspergillosis, for example. And I'd like to identify and characterize proteins that can be used for antigen antibody assays, which will help with epidemiological analysis. And lastly, I would like to explore the potential to use next-generation sequencing in the clinical setting for susceptibility testing, for example, or drug resistance monitoring. I'm going to leave you with this question because I understand that capacity is different from laboratory to laboratory. So would a serological assay be useful or a molecular assay be useful, or both of the above, or neither of the above? If you'd like to vote now. Brilliant. Okay, so 84% of you would appreciate both assay types. So as my last thought-provoking slide will be this. Could microbiological evidence be more supportive of clinical decisions when treating invasive fungal disease or suspected invasive fungal disease? In other words, without clinical evidence, could microbiological evidence, based on assays such as a real-time PCR looking at gene expression, support a probable diagnosis of invasive fungal disease? I would just like to thank the following people for their help in guidance for identifying what would be a suitable clinical diagnostic tool, and also thank Viapath for funding the next generation sequence work that I performed. So thank you. Any questions?